mentioned, my interest has been in the immunology and the immune response against parasitic helminth infections. Ever since I was a graduate student, I've been fascinated with the biology of these organisms because they obviously look really cool, and very, very large organisms are very different from the kind of parasites that you think of normally. And it's also worth pointing out that a lot of people are infected with these worms. It's estimated that about a third to a quarter of the world's population is infected with helminths. And during the course of human evolution, this number is probably closer to 70%. So if you survey wild primates in Africa, about 70% of them are infected with worms. And the point there then is that we've really co-evolved with these organisms, just as we have co-evolved with our gut microbiota. But these are not commensal organisms like the gut microbiota. These are parasites that are one of the leading causes of morbidity in poor developing countries, where the morbidity comes in often occurs when there is a range of immune response against these parasites. And so what happens is that there are some people that have a very weak immune response against these parasites, and they become super infected and have very high doses of parasite burdens, and that obviously causes problems because it's a competition for nutrients with the host. But at the other end of the spectrum, you often have immunopathology that's caused by mounting an immune response against the parasites that's too strong. And therefore, you can develop symptoms like lymphatic filariasis here that is really driven by collateral damage of the immune system against the host. Now, in the majority of the people, you have a balance between the ability to expel the parasites and the ability to tolerate having some of these parasites within you. And therefore, you can exist in a kind of asymptomatic state. Now, the problem is that even if it's just a small percentage of people that develop symptoms, a small percentage of 1 billion people is still tens and hundreds of millions of people that develop symptoms from these infections. So one of the things that I've become really interested in over the years, and it's really still very poorly understood, is why is there this variation in immune response against these worms? And this variation is the driver of morbidity of both ends. So if we can understand what drives this variation, we can better understand morbidity as well. Now, the other thing that has emerged is this concept of the relationship between these parasitic worms and the hygiene hypothesis. So the concept here is that the worms, in its interaction with us, has developed ways to regulate the immune system. And in a population that's free of parasites, this immune regulation mechanisms from the parasites has been removed. And therefore, individuals have now shifted up in terms of the strength of the immune response and become more prone to development of autoimmune diseases and other types of inflammatory diseases that we see emerge in the developed world. So I became interested in this question of the hygiene hypothesis actually when I was a postdoc at UCSF. I met an individual who was 36 years old at the time. We've all aged since then. This was quite long. It's more than now 15 years ago. And he had symptoms of ulcerative colitis and was basically refractory to high-dose steroid treatment at the time. And he decided to actually infect himself deliberately with Trichuris trichura whipworm and was able to obtain parasites from Thailand in order to be able to do that. And this was fascinating for an immunologist like me because he was basically able to put his disease into remission. And so we started monitoring him together with some gastroenterologists and were able to obtain biopsy samples from colonoscopies when his symptoms actually deteriorated, when his worm burden decreased. And also, he reinfected himself again and was able to put his disease back into remission again. So what we were able to do is basically generate a very detailed cellular and molecular portrait of events and trying to understand the mechanism of what was going on in this individual who had deliberately infected himself with worms and been able to put his disease into remission. So that was more than 15 years ago now. So over the last 5 to 10 years, we've been studying a number of healthy individuals and patients with inflammatory bowel disease, and this is the model of what we think is occurring when patients undergo colitis. So what we think happens is that you have some pro-inflammatory bacteria that are actually normally present in normal healthy individuals, but in normal healthy individuals, these are kept in check by competition with the gut microbiota and by having an intact mucus barrier. Under colitis conditions, these bacteria expand and spill out into the lumen, and also cross the intestinal epithelial cells to activate immune cells to come in to control these bacteria, including lots of neutrophils. And these are actually aerotolerant bacteria. So the oxygen gradient could well have a role to play in why they become expanded. What we think happens during helminth infection is that you have a type 2 immune response that basically stimulates goblet cells to produce a lot more mucus. And this mucus then helps to restore the barrier that has been disrupted during colitis. And what we found more recently is that this mucus-rich environment that's been stimulated by the parasites actually also alters the composition of the luminal bacteria. And they become more capable of competing against the inflammatory bacteria that can expand during colitis. So we've been using, over the last couple of years, mouse models to study in detail the mechanism by which this kind of competition can occur. And what we found is that the whipworm Trichuris, and in the case of the mouse Trichuris murus, when this infects mice, it increases the abundance of a population of bacteria called the Clostridialis bacteria, and these in turn inhibit the expansion of Bacteroides bacteria. So what we thought initially was that the worms would directly inhibit the Bacteroides bacteria, but it turns out to be an indirect route whereby the type 2 immune response that is stimulated by the worms increases mucus production in a way that promotes alterations to the ecology of the gut, and having clostridial bacteria that can now then inhibit Bacteroides that are pro-inflammatory and that drive disease in this setting. So one of the things we wanted to do is to move from these mouse model studies to look at human populations. And I've been extremely fortunate to be able to work with Yvonne Lim in the University of Malaya. I'm from Malaysia originally myself, so it was a real pleasure to be able to go back and find collaborators back home to be able to do some field studies. And Yvonne has been working with a population of indigenous Malaysians called the Orang Asli for now decades. And she's developed very close relationships with them. And these individuals live in the fringes of the jungles in Malaysia and make their living by often selling jungle products to urban dwellers. And so the settings that they live in results in many of them having helminth infections. And so we've been working with Yvonne to study this Orang Asli population in Malaysia. So the first study we did was driven by a graduate student in the lab, Su Ching, who had done a cross-sectional study on about 50 individuals from 3 different villages. And what we did was simply characterize the microbiota of the individuals that were infected by helminths and the ones that were not infected by helminths. And what we saw was a small but statistically significant increase in microbial diversity in the helminth-infected individuals compared to the helminth-negative individuals. And this was unexpected because typically infection is associated with a reduction in microbial diversity. So microbial diversity is often reduced in poor health, and so infection tends to reduce microbial diversity. So in this case, it was interesting to note that the helminth-infected individuals had greater microbial diversity than the negative individuals. So Su-Jing then embarked on a longitudinal deworming study to test this hypothesis further. So in this case, she collected stool samples from individuals before treating them with deworming medication, and then collected stool samples again after deworming to see directly what changes of the microbiota occur after deworming. And what she found was that indeed deworming reduced in a small but significant way microbial diversity in these individuals. This is I should note that this reduction in microbial diversity is nowhere near as dramatic as the difference in microbial diversity between these orang asli and, say, those of us that live in New York City and the ones that live in Kuala Lumpur, where there's a much more dramatic difference in microbial diversity when we look at different populations of people. So the effect is not as great as differences in geography. But in addition to looking at the microbiota, Su-Jing has also done a nutritional survey on these individuals to look at the contribution of the diet to the microbiota. And we've also collected blood from these individuals for RNA-seq analysis and to measure a number of clinical blood parameters before and after deworming to see which of these components really have an effect on the microbiota. And a postdoc in my lab, Meisan, developed an approach to try and quantify which of the variables that we measured had the biggest effect size on the microbiota. And we were pleased to see that helminth infection status, whether they were infected or not infected, had the biggest effect size on the variation of the microbiota, followed by the infection intensity of trichuris. And this effect was actually greater than dietary fiber consumption, which is something that's been well established to affect the microbiota, is how much dietary fiber do you consume. So based on these preliminary results, we were successful in obtaining a grant to expand on these studies. So these studies were done on a relatively small number of individuals, about 50 to 70 individuals in just one village. So now we've embarked on a larger study to look at up to 800 orang asli individuals in 4 to 8 villages and compare them to also 100 urban controls in Kuala Lumpur, and to use a similar strategy of collecting pre-treatment blood and stool samples and then followed by deworming treatment and then collection of blood and stool samples after that as well. And one of the kind of the main questions that we want to address in this study is really to look at, because these individuals, we will also be able to genotype them for SNP variants. And the question we are really interested in is really to determine the relative genetic microbiota and immune contributors towards the variations that you see in helminth infection burden, and also the efficacy of deworming. So one of the things we noticed was that, trichuris infection, the deworming treatment is not 100% effective, unlike for hookworm and ascaris. And so there's quite a lot of variation in the deworming efficacy in this population. And one of the things we noticed was that the people that were successfully dewormed compared to the people that were not dewormed successfully had differences in microbiota compositions. So we didn't have enough individuals to really look at that in detail, but we hope that with this study we could examine the effects of the microbiota on the efficacy of the deworming treatment as well. And also susceptibility to becoming reinfected. So individuals will often become reinfected, and whether or not this is because of the genetic or the immune state of these individuals is not well understood. And this work is done in collaboration with Yvonne Lim, as I mentioned, and Ken Cadwell in the Medical Center, and also Rich Bonneau, who has been helping us on the computational biology aspects of this work. Just one more thing that to mention is actually one of the things that Ken in particular is interested in is actually looking at the fecal microbiota from these individuals to see if we can isolate specific bacteria through passage in germ-free mice that are more potent at inducing regulatory T cells. In a sense, can we isolate actually more potent probiotic strains from these individuals that could be used to treat inflammatory diseases? So I think this just kind of brings me back to this slide, and where I want to make the point that we have still a very poor understanding of why there is this variation in the immune response against helminth infections. Indeed, we have a very poor understanding of immune variation as a whole, not just to helminth infections, to all kinds of infections. In fact, for cancer immunotherapy, for example, we know that some people respond and some people don't. And so what we want to understand better is the relative contributions of genetic variation and environmental variation towards these variations in the immune responses, and this is an important determinant of morbidity during helminth infections, but also could address the treatment of inflammatory diseases. And with that, I'd just like to thank actually the research subject, the first person I pointed out to you that I met, because we wouldn't have embarked on all these translational research projects if I had not met him. I was predominantly a mouse immunologist and studying mouse models of immune response, and now we've embarked on all these field studies, and that's kind of triggered by this interaction. I've had great collaborations on the West Coast as well as on the East Coast, and also with the University of Malaya, and with that I think I'll just stop here and